Should we start? Okay, so welcome to the CGSI long course. Without any authority, I'm welcoming you all to the course. This is my third year at CGSI, giving 2 talks a year, so it's my 6th talk. And I don't know how many people here have heard all the previous 5. Maybe Zar is the only one, but or maybe Bogdan as well. But there was actually 0 overlap in topics between my previous 5 talks, and this was sort of taking into account that people repeatedly hear it, which may not be accurate, but also to challenge myself to talk about a different thing every time. So this is actually completing the sequence of 6 talks on completely unrelated topics. And given that sort of biology-related stuff is actually not a majority of the work we do in my group, then I'm kind of running out of topics. So I'm down to talking about something that I don't really know that much about.
Are you saying next year you'll be repeating your first talk?
That might well be the case. The bootstrap tutorial was my first talk. Okay, but it's work. It's something that I actually think is very interesting. So I do want to talk about it, and it's a project that my student Aya Vituri and I have been working on for a long time, without that much success, but encountering all kinds of interesting things along the way. So I'll talk about it, and everyone is welcome to ask questions. If I can answer them, I will. If not, we have a week. We'll set a time and read up and figure out the answers together. Okay, so this is a talk about speciation and speciation without isolation and without adaptation. So what's speciation? So there's all kinds of definitions. One very common one is the reproductive isolation between 2 taxa. So once they cannot interbreed, they are 2 different species. So modern humans and Neanderthals are probably not 2 different species according to this definition. But we can also say that like pornography, I know speciation when I see it, right? So we know what it is, but it generally means that the species cannot interbreed once they have speciated. And there's a traditional view of what drives speciation. Basically, the speciation is driven by isolation and adaptation. So a species somehow splits into 2 groups. It might be a geographic split, or it might be actually some social drift or something else. And once those 2 distinct groups, they start diverging from each other by adapting to their environment or to the niche within the environment. And that adaptation is driving them further apart until they are no longer genetically matching and cannot interbreed. And one interesting question is are these 2 elements of isolation and adaptation really required for speciation? Can speciation happen without adaptation and without isolation? And one thing we might think about, at least as a mental exercise, is our species today, which is a huge species, and it has small effective population size, but huge actual population size. It's fully interbreeding, as far as we know. So, and not only is it fully interbreeding, it's also practicing its ability to fully interbreed, right? Because people go to UCLA and meet other people from all over the world, and whatever transpires happens, and then there's really no isolation within the population. So these 2 words are going to play a role later. So sympatric means completely random mating, and topopatric, which might be more appropriate description of the humans, is that I tend to marry the girl next door, but once in a while I go to UCLA and meet people from all over the world, right? So with high probability I choose my mate close by, but with some probability it can be anyone in the world. So that's basically a topopatric model of how the species breeds. And the other thing we may or may not agree with is that there's no real adaptation going on within modern humans, because basically there's a public health system and everyone gets to breed. So we might argue there's some social selection going on because some populations breed more than others and so on. But to first order, we might argue that there's no real either isolation or adaptation going on. So one question is, in such a setting, is there ever going to occur speciation? Okay, so if we, as a mental exercise, assume that the situation stays the same for the next 10 million years and we come back, are we going to see a single species, or is it somehow inevitable that there will be multiple human species in that setting? So one very interesting aspect of this thought exercise is this well-known and not new at all model called the Bateson-Dobzhansky-Muller model. And it basically says that mutations can, and actually it's a common occurrence that mutations are neutral on their own, but they are lethal in tandem. So we are now not talking about positive selection, negative, it's perfect negative selection once the two mutations meet, and on their own they are neutral, okay? So just as an example, let's say we have only two loci and the population starts as everybody being big A, big B, homozygous, and the neutrality says that if we have a mutation from big B to small b, we still have perfect fitness, or if we have a mutation from big A to small a, we have perfect fitness. But the genotype small b is actually inviable and cannot be born. Okay, and we may assign different fitnesses to the heterozygous, but we just think about the homozygous because we think about things drifting and then fixing by drift, and then something happens and you can no longer have babies. Okay, and another option might be actually that big A small b and small a small b are neutral, but small a big B is inviable, because this can also happen, right? So we have small b happening and fixing, and then in the people who have small b, we have small a happening and fixing, and then they cannot breed with people who have the big B, the original big B. Okay, so either of these is actually consistent with the model. And this is by no means a new idea that this can happen. And in fact, I think already many years ago, it was proven in the lab that such situations do occur, where each mutation is neutral on its own, but together they are lethal. Okay, so let's just take this as our model, the BDM. The only thing that's going on is the BDM. So the claim is that in some sense, BDM means inevitable speciation. Why? Because, so let's take the situation where small b is lethal. Then there's some chance of big A to small a happening and some chance of big B to small b happening in different individuals. And then there's some, maybe very small but bigger than zero chance that they would spread throughout two halves of the population and fix, this one would fix here and this one would fix here, and then these two halves would not be interbreeding, right? So it's possible. I mean, it's extremely unlikely, but it has this setting that I described has probability bigger than zero, right? And what do we know about stochastic processes? That if we wait long enough, anything that can happen with small probability will eventually happen. So in some sense, this is a very minor conclusion, but it's still interesting that if nothing else happens to disrupt this process, like extinction or a speciation of the old part using isolation or adaptation, if nothing else happens, then this alone is enough to cause speciation eventually. Okay? Nothing else needs to happen for speciation to happen. So the weak conclusion is that if we wait long enough and nothing else happens, there would be speciation based on this alone. But now we go back to what is a speciation. So how big does the speciated group has to be, and how long does the speciation of more than one species has to persist, so we actually call it a speciation. So these things might not behave the way we think of a speciation. Yeah.
Right, but if that was the case, this is extremely unlikely, because you would need to have the people that have kind of both to kind of disappear.
Yes, right? yes. But so let's say I choose some arbitrary point and divide my population in two halves in my mind, yes, along this arbitrary point. Now I give you a bigger than zero probability event that this mutation would happen here and this mutation would happen here, and then they would each fix in its half. It can happen. It's super unlikely, but it can happen. Okay? So that just means that from this very simple probability concept, it will happen eventually. But that doesn't mean that this is what we should be interested in when we're investigating speciation, just that it's possible. It's possible that there's no adaptation and no isolation, and speciation happens using this BDM model. Okay, so if we want to try to calculate, or I'll talk a bit about why we can't really calculate, but at least simulate this kind of thing, then we need to make this probability a bit higher, because otherwise we would never see it in simulation either. So, and also we might ask ourselves, what are other assumptions that we might assume when we think, say, about modern humans? Okay, so there's two additional assumptions that we think are kind of reasonable. So it just takes too long and we can simulate it. So what other assumptions can we make? So the first one I mentioned already, this idea of a local preference. Okay, so if we also assume local preference, it's going to significantly accelerate this kind of behavior, right? Because once a mutation happens, it spreads in the neighborhood, and that mutation happens there and spreads in that neighborhood. Then eventually the two neighborhoods become two distinct species. So that local behavior, which is not, we're not assuming isolation, we're assuming sort of continuous isolation, right? Because I prefer my neighbors. And the other one that's very interesting, and many papers assume it, is this assumption of assortative mating. And assortative mating, we may argue for a long time whether or not it makes sense in human, but it basically says that I tend to select mates that are not only or not at all geographically close to me, but genetically similar to me. Okay, so now we might think of so far everything we thought about was completely neutral, we had no traits, but now we might think that the way I look is determined by my genetics, and with the Oedipal complex and so on, I tend to prefer mates who look like me. So, so that would be sexual selection, or it's called assortative mating in this context. Okay, so this will also, of course, accelerate speciation of this kind, because once I select people who are like me, I tend to select people who have the same mutations as me, and we cluster, and the mutation spreads in the group of similar individuals, right? But note that we are not assuming in any way so far that anything else is determining cross-fertility other than these BDM pairs. So being genetically dissimilar, the only mechanism by which it makes reduces fertility is by these pairwise contradictions. And the BDM model actually thinks about more than pairs. We can think about triplets, quadruplets, that the four mutations together create inviability, but it's really the same concept. So we can start by thinking about pairs only. So let's try to simulate this. So the first simulation I'm going to show you is using assortative mating only. So basically only preferring mates who have fewer genetic differences. And this is just to demonstrate that interesting things of the speciation kind happen when you simulate this. It's a small population, 1,000. And the reason is that our simulation is so far not very efficient. So if we want to run for 100,000 generations, we have to do it with a relatively small population. But we see this effect of basically the number of non-interbreeding groups being 1 and then jumping to 2, and then going back to 1, meaning one of them goes extinct, and we're back to 1, and at some point we even go up to 3. But there's clearly no, in this simulation, no really stable speciation going on. But there is this kind of behavior that we expect from the BDM happens in this setting that has no isolation of any sort. It's sympatric and no adaptation, only assortative mating.
Didn't you forbid extinction in your model?
Extinction of the entire species, yes. The bottom line here is one species. So you always have at least one species, because we're in a fixed population size. And then the question is how many groups that cannot interbreed between them exist in the population. So the fact that it appears and then disappears means it's relatively small groups that are not random and not interbreeding with the other groups, and then they go extinct after a while, okay?
And what is 3 in this context?
3 means there's 3 groups that no 2 of them can interbreed.
You
So
have multiple mutations of this sort.
yes, you have like I mean, this is the most vanilla thing you can think of. It's an infinite infinite alleles model. So each mutation is unique, and then each pair of mutations has some fixed probability 0.1 of being a BDM pair. We can take it a bit further and think about now adding this topopatric, this local selection effect. And this graph contains more information than we need now. I'll show it again later. But basically, the black line, so this is now averaged across many runs. The previous one was just one single run. This is averaged across many runs. The black one is the average number of species or the median number of species across many simulations in just BDM with topopatry. So a preference for local mate selection and nothing else going on. And we see this behavior that after a while we get multiple species, and at least in this simulation or in this set of simulations, it seems that that 3 is maybe a number that in this simulation setting, now we have a bigger population size, 10,000, and we see that we accumulate species. And this also shows in blue the simulation that also adds assortative mating, and it has this very interesting behavior that the number of species goes up much more quickly than without the assortative, without assortative mating, because this preference for people who are like me actually very much accelerates this accumulation of mutations, but for reasons that I'm not sure what they are, it doesn't seem to keep increasing, but actually stabilizes at about 3 as well.
By the way, Saharon, can you prove, or if not prove, can you show some convergence here of this?
Okay, so I know people expect me to have formulas and mathematical results in my talk, but
even by simulation.
So we have to improve our simulation results, but we so far, I mean, we can't go much further beyond this if we want to run. This took months to run this thing. Okay, but there's a lot of room for improvement. I'm going to show some mathematical calculations that are not ours. Basically, my argument is going to be that within what we want to simulate, it's impossible to do. We have no idea how to do mathematical calculations. We have to add assumptions which we don't like to do mathematical calculations. I'll show that. I'll show some work by other people with additional assumptions. Okay, so what we've concluded so far is that the just BDM, or there are other models that we think about that diversity basically decreases fertility. So for example, homologous recombination we know is important for error correction and so on, and if the species is very diverse, then the rate of homologous recombination decreases, right? Because there's less matches between the 2 copies of the genome for the individual, so there's less recombination. So we might think about a different model where increased diversity decreases recombination, and that decreases fertility of the species. But that's a very different model. That's actually going to be a model that's much easier to analyze. Why? Because in that model, you just pay for being diverse. In the BDM model, you don't pay for being different. There's specific pairs of mutations that would cause speciation. Right? The speciation is not caused by an integral over the differences, but is caused by a specific pair of mutations that causes a BDM. So that's kind of a hint into the mathematical difficulty of this model, that you have to identify each mutation has to be identified, and then you have to identify a pair that creates in your simulation that creates a mismatch, and then whenever this pair occurs, there's a mismatch. But two individuals might be very different and completely cross-fertile, or might have just this difference and be non-interbreeding. Okay, so not surprisingly, things like topopatry and assortative mating speed the speciations up, and we might or might not ask ourselves whether this is some hint about the process that modern humans might go through if they persist as a big interbreeding species without real selection.
I was asked a question. So when you say the second bullet point that the local preference increases the speed of getting new species, is it also the case that these species have fewer individuals? Is there like somewhere in the model an assumption of how many individuals are in this species?
Nothing we've done looks at how many individuals, but I don't see any reason why the species would be smaller in this setting.
Is there like more local?
Yeah, they are local, but we have one species forming in the US and one in China, and they expand locally. So you could eventually.
Keeping the total number of individuals in your simulations
Yes,
is to like
yes.
10,000.
Yes, so they expand locally, and then by the time they meet, there's 5,000 of these and 5,000 of these. It's possible. I'm not sure if that. It's a good question about the simulations. I think Aya did a lot of statistics on the size of the species, but I don't have it here.
So how do you define a different species? Is it that the incompatibility loci have to become fixed in the different groups?
Right, but actually we have individuals, right? So two individuals who meet and they have a BDM, that's a failure to breed, okay? And then you have to count these failures, and after you go above some threshold, you have this group of people who can't interbreed with this group of people, okay? So again, I think there's a lot of sort of degrees of freedom in how you exactly define speciation.
Is it a graph of, do you define some graph with components, with connected components?
You should do it. I'm not sure exactly how. So the question is, so once you have these two mutations that are BDM, so the structure of the graph is very similar. It's very simple. Everyone who has this mutation fixed is non-interbreeding with everyone. Everyone who's homozygous for 1 is non-interbreeding with everyone who's homozygous for 2. Right? So, I mean, in terms of graph, it has very simple structure. It's just a question of setting a threshold on how big these groups are. And yeah, and I'm not sure the answer to that. But we see that, for example, in our simulation, that things stabilize. Once they stabilize, it's clear that these groups are pretty big, because otherwise they would be going in and out. Okay? Okay, so let's satisfy our need for some calculations. So here's a different model, I think pretty famous model within this community, that says, that sort of relaxes some of the assumptions of the traditional model. This is a model where there's an ancestral population and a derived population, and there's gene flow between the populations. But we are ignoring the diversity within each population. We're basically assuming that the populations are homogeneous within them, meaning we know where the speciation is going to happen. The speciation is going to happen on this line separating the two populations, right? And now we can do some calculations in this model. Basically, we have to say when will a mutation that's like a BDM contradicting separate this group from that group. So this is actually pretty easy. The important assumption here is that actually that the groups are homogeneous within. So let's try to follow this calculation. So let's denote the mutation rate by this nu. And M is the gene flow between the populations. Q is basically the BDM probability. And D is the current genetic distance between the populations, actually not used here, because what we want to do is we want to sort of estimate, understand the behavior of what the genetic distance will be when the speciation occurs. So when this population and this population have a BDM contradiction, so how many mutations and how much time. Okay, so, and there's all kinds of nice calculations we can do with it. Notice that there's basically a geometric distribution here. We think about mutations happening and fixing, and we need just a pair to be contradicting, and there's approximately k-square pairs, and these calculations basically follow. There's a lot more calculations like this that we can do, be more specific, calculate expectations or approximate expectations, variance, and so on, but this is the kind of behavior we get. We get that we basically expect about square root of 2 over q mutations until we see speciation to happen and fix in each subpopulation, and from that we can derive an expression for the approximate time. Okay, and
Why was 1 over V there?
1 over, how do I go up here? 1 over Q.
No, 1 over V.
T.
Maybe it's not V.
Yeah, so, no, this is new, yes. Yeah, so this calculation, I don't remember where it comes from. This is basically just the, I guess this has to be the time for fixation of K mutations, T. It's kind of population genetic calculations that I don't remember the details of. But the first one is basically just using the geometric.
Yeah.
Okay, so the calculation I showed you so far assume that the populations are homogeneous within and the speciation will occur exactly on the split line. So basically we are bringing back isolation into our model, which is not what we want to do. And one question we might ask ourselves is it actually, so under what we want to do, do these assumptions keep the behavior similar, or do they change the behavior completely? Right? So, for example, under BDM, it might happen that the split actually happens, even in this model, the split might actually happen within one of the populations rather than between the two populations, right? Because actually within each population, we also have genetic diversity, and it might be that the mutation fixes by chance, by drift in some part of the population and the other one in the other part, and we have a speciation. And also the whole idea of subpopulations is not how we want to think about it. At least when, say, we think about modern humans, it's not really, well, okay, we might argue that, okay, so I won't argue this point too much. Okay, so when we look over what people have done in this area, where we can find many different works, but we see that all of them have an additional assumption. So, for example, this set of papers by the same group, they basically simulate haploid. They are interested in, this is specific papers interested in speciation, and speciation of the general same kind we're looking at, right? So, so not really based on isolation and adaptation, but these people do haploid populations. And notice that in a haploid population, the kind of BDM speciation is impossible without assortative mating, because with haploid individuals, this contradiction is impossible. As long as the two parents are viable, you can have viable children. And thanks to Sagi for pointing that to us quite a few years ago when we started working on this project. I don't know if you remember you did that, but you did. The fact that BDM speciation is impossible in haploid populations, in haploid species, only in diploid. So they need assortative mating to get any kind of. So the assortative mating gives you now you prefer people, you prefer individuals who are similar to you. Then this creates this drift of subpopulation. And one thing I want to mention is that this is not. This is relatively recent and not esoteric work. At least one of these papers is in Nature, so so people are interested in the kind of behavior, speciation behavior that arises out of these models. Another line of work is assuming that populations are diploid, but assuming that the fertility is a decreasing function of genetic difference. And this is what we call the threshold model, even though it's not necessarily threshold, it might just be a monotone function. But this is fundamentally different than what we're assuming, right? Because in our model, you might be speciated with one difference, and you might be interbreeding with a million differences, as long as there's no specific BDM pair that's the distance between the populations. So, so this threshold is a common assumption, and why? Because A, it speeds speciation, and B, it helps in analyzing it mathematically. Okay, and what this is usually used for is to examine how speciation processes happen, and for example, how long to speciation, how many species, dynamics over time on grids, and so on. Okay, so from all of this, we can build this taxonomy, which is kind of a major aspect of what we did, of the kind of assumptions that studies use, and kind of justify our project by saying that, okay, we can look at haploid or diploid populations. Within haploid populations, we must assume this assortative mating and threshold behavior to get any kind of speciation. Without diploid, we might take the threshold route, or we might say there's no threshold, there's this random process with identified mutations where there's specific BDM pairs. And the point we try to make is that this is actually empty from previous work. This region that looks at how speciation happens without either threshold or being haploid and so on. So what we set out to do is to simulate, try to select a simulation setting where A, we can run for long enough to get results, and B is in some sense representative of of realistic scenarios, and then see the behavior of the speciation under the different models. So there's lots of simulation details, and I don't think I'll really go over them. So points that should be mentioned here. We have an infinite allele model, we have full recombination, and we are either diploid or haploid depending on what we're simulating. And we have 10,000 individuals, and they're all genetically identical to start with, which is also kind of similar to humans, given we have I guess 10,000 is representative of current effective population size of humans. So this is going to end where we are now, but it starts being homogeneous. So relative to our actual population size, our effective population size is tiny. In that sense, it's realistic. Okay, and the last point is important. To simulate BDM, you have to keep the identity of all mutations, and each pair of mutations that meet have to go through a draw whether or not they are BDM incompatible, and that has to be maintained for all future meetings of these mutations, right? And mutations that are not BDM incompatible will remain not incompatible. That's the thing that's unique about that. You cannot just look at genetic distances. Okay, this is the graph we already saw. So this is basically the diploid models. And we see the two graphs that we saw before, the black and the blue. And we also have here the two models that have threshold, so that have this effect of decreasing fertility with increasing diversity. And we see that these actually have delayed, this is the purple and the red, and we see that these actually have delayed speciation. And this is not surprising either, right? Because if you just care about the distances, then basically all mutations are being selected against, right? Every mutation that happens makes you less fertile, and the mutations are being selected against. But we see this different behavior that once we also add assortative mating, so that's preferring the ones who are similar to you. At some point, the graph starts to explode, and without assortative mating, there's no speciation going on. Right, so we see that behavior here. We extended the length of the graph to 150,000 generations. We see this explosion of species when we also have assortative mating, whereas the speciation process is frozen when you have only the threshold behavior without assortative mating. Finally, we might look at the haploid cases as well. And the haploid cases just explode in our simulation. And this is, when you think about it a little bit, it's not surprising either. In particular, if we have haploid and assortative mating and threshold, the number of species explodes. Okay, and this is just for the statistically oriented, to just make sure that the differences we see are significant. We do some permutation testing to make sure that we see actual significant difference between the settings, and basically all interesting differences are very significant. Maybe interesting that is not significant is this one, that the addition of can we see we don't see the cursor? Only I see the cursor. the third line, BDM assortative mating compared to BDM, we see not a very big difference, meaning, or not very significant, meaning in some sense the addition of assortative mating to the regular BDM doesn't make a big difference. But the other models on the other nodes of the graph have very different behavior than the BDM. Okay, so maybe not very exciting conclusion, but one conclusion is that the assumptions make a big difference, the assumptions under which you're simulating. And the BDM, at least BDM and BDM with assortative mating, if you lump them together, have a unique behavior. So if we care about this, we cannot really draw conclusion about speciation behavior from simulating other things. Okay, so overall, so in some sense, speciation without isolation and adaptation is possible, for example, due to BDM, but we can think of other mechanisms like homologous recombination. The assumptions make a big difference. And if we try to infer conclusions about the future of humans, it probably makes sense to make as few assumptions as possible. And at least in our efforts, we have no way of doing analytical calculations within this model, only to simulate. Okay, thanks. Okay, good.
Questions? I don't know if it's a question, kind of a thought. Is there a sense in trying to define a model so none of the existing models that maybe can be theoretically analyzed is a good, I don't know, provide good bounds for the PDM, but maybe there is a model that is not identical to BDM, but like in your simulations you could show that it's not significantly different and maybe can be.
Yeah, it's a good question. I don't know the answer to it because, I mean, even if you get that this for this specific simulation, something is not that different. It depends on the simulation. We spent a long time, years, trying to do math in this model, and we were basically only able to do math in a model that looks at distances rather than specific pairs of mutations. In that you can do nice math. But at some point, maybe it was wrong, but we decided that it's not really the relevant mathematical model.
So I have a sort of maybe conceptual question. So you said that if I assume this and assume this, and we are in not exactly in that project, but in a similar, where you really cannot do rigorous math. And I say to my student, cut in the fresh, in the flesh. I mean, like, do all the bad assumptions just to get something. And I want to hear your opinion.
Well, I don't know. It might be a good idea for publishing, but for me, I want to do things that I feel are kind of. I mean, I've been thinking about this for over a decade in the context of humans, right? So I'm kind of curious about doing something that's it's not going to be realistic, but that is interesting to think about in the context of species like modern humans. So in that respect, I think that the settings where we can do math are not interesting. So we ended up with, so we wrote a paper, and it's basically just showing what I showed here, which is not, it's not really what I hoped for, right? Because our focus is on showing the difference between making different assumptions rather than on the conclusions. Because if you pick a specific simulation setting, especially given how limited what we can do is, and you get some conclusions, then I mean, it's hard for me to argue that this is actually relevant to a specific species that we're interested in. Someone else might be able to argue that and try to publish it in Nature. So basically what we're able to show is the different behavior based on different assumptions.
So in terms of the set of parameters that underlie the model,
Yeah.
do you have a sense of which ones are well measured based on existing studies and which ones a lot of uncertainty
Well, okay, so if we agree, which we don't have to, that the motivating species is humans, then we have to start from a 7 billion population size, and at that point, you know. So I think you didn't ask what are the parameters affecting the, but you asked what are realistic parameters. So I think 7 billion is a non-starter for anything we do. So in that sense, we are already sort of, until we improve our computing skills and maybe our clusters, we are already detached from our real motivating application, in my view. The other thing is, if you want this thing to work, you probably need a very large population, right, with realistic parameters, because you need for these things to appear in different places and be able to spread before they meet, because once they meet, there's major disadvantage for people who carry these mutations. But I don't know the answer beyond that.
Not exactly about speciation, but assortative mating. So the data shows that there is a non-monotone behavior. People don't marry their sisters and brothers, but it's like second or third cousins.
Uh-huh.
So you have, so the probability goes up and then goes down. It's
Okay.
genetic distance. So either, do you know if any of these models can explain, or is this?
No, but I mean, so, but then maybe it goes up again when you get to really different, because otherwise I don't think it's a major concern, because you have very few brothers and sisters, right? So basically you can assume it's monotone with the, right, with the, because you have many more people who are on the order of belonging to the same subpopulation, having the same color and the same, than brothers and sisters. So I don't know, but I think if you assume it, it makes sense to think of it as monotone.
Yeah, I think it's interesting to think about human evolution on like 100,000 generation scale, but sort of the factor of BDM models is this assumption that in tandem a mutation is going to be lethal, and I wonder if you could take into account sort of reduced penetrance of that lethality, such as instead of having reproductive isolation where you just can't have an offspring, sort of more hybrid inviability, where an offspring will survive and maybe exposure during early life before reproductive age could affect based on sort of the genetic.
Yeah, I think it's a good idea, but probably people with more knowledge in population genetics and bigger clusters would be required to work on that. Okay, thanks.